Hello dear students, good morning. Today we are going to learn about euglenophytes. Okay, so let us start. What is euglenophytes? So I will tell you that euglenoids lives in, they are a type of organisms. So generally they lives in fresh water and constitute very much interesting group of organisms. Okay, so member of this class clearly illustrate impossibility of distinguishing plants from animal among the Protistas. Okay, so about 40 genera of euglenoids have the chloroplast and are fully autotrophic. The others are without chloroplast and ingested their food and are heterotrophs. Okay, some of the euglenoids like euglena that have chloroplast lose it is and thus become heterotrophic. So if the organism is kept in the dark, right, so again if these are transferred to light they become green, means with chloroplast, okay, within few hours. So for example this types of animals or organisms we having like euglena, right, yes, Paranima, Fica, Euthripita. So in this way we having lots of examples. Now, what is euglena? Okay, so euglena is the most common and typical example of euglenophyta. It is the largest uniflagellate found in freshwater pond and ditches, especially abundant in water bodies which are rich in nitrogenous organic matter like urine and feces of the animals. See, you can see this is the picture of euglena showing cell structure. You can see here this is the flagella. This long thread like structure is known as flagella. Okay, so let us talk about the structure of euglena. So you can see by the help of this figure, euglena is a spindle shape about 50 to 100 mu in length. It helps a whip-like flagellum which emerges by two roots from the bottom of the reservoirs. Okay, it helps in locomotion. It is contractile vacuole attached to one side of the reservoir which maintain water balance in the ambulance. Okay, now near the reservoir of the opposite side of the contractile vacuole a photosensitive eye spot is there, you can see, or stigma is present. Okay, so stigma is also present. Now food reserve are present by paraphylum bodies. Okay, so we can say a carbohydrate similarly to glycogen. Okay, so euglenoids are without cell wall but have a flexible or elastic pencil which is made up of protein. Now it allows their shape to change. They can change their shape also. Now let us talk about the modes of reproduction in euglena. So, euglena and other euglenophytes reproduce by asexual mode of method. Okay, means they generally use asexual methods only. Now, sexual reproduction is not reported in euglenoids. Okay, so basically we having a asexual reproduction in euglena. So let us see asexual reproduction. So it is done by binary fission. So binary fission means asexual reproduction takes place by means of longitudinal binary fission which start from the flagella end. Okay, and the flagellum duplicate first and then along the nucleus division, that is mitotic. I told you that if there is a nucleus division then it is a mitotic division. Okay, then the cell divided longitudinally into two daughter euglenoid. Now, so I can also show you the picture. Here you can see the longitudinal fission in euglena. Okay, now next we having by cyst formation. So when the water in pools dries up, so it takes place a protective measure to tide over unfavorable condition such as lack of food and oxygen, drought and extensive heat, and etc. Now cyst wall is secreted in the form of thick spherical gelatinous covering that composed of carbohydrates. Okay, when this condition become favorable the animal become active and emerge from the cyst to resume its normal free swimming like euglena. Okay, so this is the reproduction found in euglena. Now next we having slime mold. Okay, so let us know about slime mold. So they have more characteristics of protozoans, but due to similarities with fungi, these are also called as fungal animal. Okay, so the distinguishing feature of such organisms is that there is a vegetative body is a necked mass of protoplasm called plasmodium. Okay, so a slime yes, viscous membrane surrounded it, and we can say they used bacteria and microorganisms as their food, and they shows a holozoic nutrition also. So due to the presence of slimy covering, it appears like slimy mash, and due to which it is called a slime mold. Clear? Now the slime mold exists in two forms, so that is cellular slime mold, and we having next is a cellular slime mold. So let us see first what is cellular slime mold. Yes, so the cellular slime mold are represented by unicellulated haploid amoeba like necked cells covered by the plasma membrane. Okay, so when we will talk about the life cycle of cellular slime mold, so our knowledge about cellular slime mold is based on our studies, or we can say the Dicatostylum and Dizochridium. So its life cycle start with the germination of haploid, that is N. Haploid means single number of a cell, that is N. So spores having a cellulus and cell wall. Each spores produce a necked amoeba. Clear? Now sexual reproduction in cellular slime mold is conversial. Okay, so I just wanted to show you the life cycle, the cycle of a cellular slime mold. You can see this is the life cycle. So see here, this is a point, it is written here, right? So that is known as sporocarp. Okay, so that sporocarp will change into a spore, and after the spore formation it used to germinate, means germination takes place, and after the germination it is converted into amoeba. Okay. So when you will see, I am talking about asexual reproduction. Okay, so after amoeba formation they will change in the aggregation. Okay, you can see they will attached, they fully attached. Okay, so aggregation, and at last they will form yes, pseudoplasmodium. You can see in the F point, right? So this is about asexual reproduction. Now if we will talk about sexual reproduction, that means after the germination this will convert it into amoeba. Okay, so amoeba, so this amoeba without aggregation, means without going closer or attached a single copy, what they will do, they will form a plasmogamy. Okay, and these plasmogamy will convert it into cryptogamy. Okay, and after the formation of cryptogamy, what happens at the last, zygote is formed and meiosis division takes place. Okay, means after the zygote formation meiosis process takes place, and that meiosis process will form the aggregation, and at last yes, pseudoplasmodium is formed. Clear? So this is all about the cellular slime mold, means both the cycles, asexual as well as a sexual reproduction. Now let us talk about a cellular slime mold. Okay, so the acellular slime mold are represented by yes, Physarum and steminoids. Okay, so all the diploid nuclei, that is continue dividing by mitosis in a synchronous manner as the protoplasm steams over. Okay, so we will in this also we will talk about the life cycle. So the life cycle of acellular slime molds started with the germination of spore having distinct cell wall, which produce 1 to 4 non-flagellated swarm cells. Okay, so let us see what is swarm cells. So come to this life cycle. Yes, so this is the life cycle of a cellular slime mold. You can see in the first point there is a spores. Okay, then they will do the germination process and they started to germinate. After the germination, what will happen, the cell division will start. So you can see one side there is a cell division, and on the other hand you can see the right hand side there is a swarm cell is formed. This is known as swarm cell. Okay, which is means inside from the spore, after the germinating this forms. Okay, So after the cell division, what will happen? These swarm cells means the flagella takes place. Flagella, you can see here a thread like structure, that is known as flagella. Okay, so these flagella will take place, and they used to they used to increase their size. Now plasmogamy stage will takes place, and they will take a little shape like amoeba. Okay, after the plasmogamy, you all know that cryptogamy stage takes place, and cryptogamy is known as almost the formation of zygote, right? So after the zygote formation, young plasmodium is formed. Then that young plasmodium will become older plasmogamy, and after becoming old plasmogamy, it beginning of the fructing. Okay, it beginning in the form of the fructing. You can see in the K point. Yes, so you can see the beginning of, yes, fruiting. Clear? So this is the life cycle of a cellular slime mold. Okay, now next we having protozoans protista, that is protozoa. So let us see what is protozoans. So it is first studied by the Leeuwenhoek in the year 1677. The name protozoa was coined by Goldfuss in 1871. Protozoan were treated as first animal before the discovery of bacteria and other moneras. So therefore they are named as protozoa. Okay, so protozoa, as you all know, that protos means first and zoans means animal. So it is the largest group, which included about 50,000 species. Let us see some characters of protozoa. So means they are generally microscopic and unicellular. They are heterotrophs. They show solitary but sometimes become colonial. Okay, they are generally aquatic and found in a moist place. So in this way we having many characteristics of protozoans, you can see. Now let us talk about the classification of protozoa. So on the basis of five kingdom classification, the protozoa and protista have been divided into four phyla. On the basis of their locomotion and presence of the locomotory organ, these are like Rhizopoda, second we having Mastigophora, Sporozoa, third, fourth is Ciliata. Okay, so a brief description of all four phyla has been present. You can see the classification. Okay, so this is the classification. Now let us talk about little bit about these stages or classification. Okay, now Rhizopoda. So what is Rhizopoda? So they are irregular in shape like amoeba. Amoeba also not having any defined shape, right? Locomotion by means of pseudopodia, and pellicilla is not found. Pellicle is not found. Now nutrition is holozoic. Clear? You can see here in this structure. Yes. Now next we will talk about the Mastigophora. Okay, so Mastigophora, this is one or more flagella are found for the locomotion, and body is covered by the pellicle. The cell body has definite shape. Okay, so in this way we having many characteristics, you can see. Now trypanosoma and Leishmania. So trypanosoma, let us talk about. So it is a zooflagellate parasite which causes an African sleeping sickness, as we already aware by this word sleeping sickness. So in human being, and also it is found in western and central part of Africa. Okay, and the parasite is transmitted by blood sucking testis fly, right? Yes. Now, next we having Leishmania. So, what is Leishmania? It is a flagellate protozoan protista which causes Kala Azar. This we have already studied in our last class, in 10th class, right? Kala Azar and dum fever. So, it means black sickness. Okay. The parasite is transmitted by sandy fly, and we can say dogs and cat functions are the receiver host. Generally it takes place in dogs and cats. Okay, now sporozoa. So sporozoa means all the parasites, they are endoparasitic. Now pellicles is found surrounding the body, locomotory organ are not found, contractile vacuoles are not found. In this way we having many things. Now let us talk about Plasmodium, that is malaria parasite. This is very most important topic. So Plasmodium malaria parasite. Plasmodium is a human blood parasite, right? It causes malaria fever. It completes its life cycle in two hosts, that is man and mosquito. Female Anopheles is a vector of Plasmodium. The primary host is man. So when the parasite maintains amoeboid stage in the RBCs and later produce gametocytes. When female Anopheles feeds on human blood, gametocytes enters to the stomach of the mosquito and develop into gametes. After the fusion of gametes, zygote is formed, which changes into cyst after entering in a stomach wall. You can see the structure. This is the life cycle of full Plasmodium. Okay. So the cyst produce sporozoites which reaches salivary gland of the mosquito. When such an infected female Anopheles sucks blood of a healthy man, it transfers sporozoites to repeat the amoeboid stage again. Clear? So this is the life cycle and the process of our malaria parasite. Okay. So now let us talk about ciliate. So its body is covered by the pellicle. Now cilia are present all over the surface of the body. Now contractile vacuoles are found. So you can see we having many things you can read from here. Now let us talk about the paramecium. Okay, paramecium, that is sleeper animalcule. The most common freshwater ciliated is a paramecium. So it is a sleepy shaped, thus it is known as slipper. Animalcule. It is an active predator and feed on the practices sucks into a mouth like cavity cytosome. The feeding water current is created by the beating of cilia or oral grooves. Unicellular ciliates have a large number of specialized organelles than any other eukaryotes. The function of these organelles are equivalent to the mouth and digestive tract, anus, kidney, muscles, skin and sense organs of a high animal. The cilia provide locomotion superior to that produced by flagella or pseudopodia. Ciliates can be removed at the speed of 2 mm per second, and they have defined organelles like trichocyst. Yes, and these small dark dirt-like structure are ejected with the force to stumpy. Now, let us talk about the reproduction in paramecium. So, under the favorable condition, paramecium reproduce asexually by transverse binary fission. It also undergoes periodically various kinds of curricle sexual reproduction, such as conjugation, autogamous, cryptogamous. So, in this way, many types. Okay, you can see also here. This is the different stages of conjugation of paramecium. So now next we having the Protestant symbiotic forms. Okay, so in symbiotic association both species are benefited and neither species is harmed. Some Protista lead symbiotic life such as unicellular algae, right? Yes, like zoochloral. So these live symbiotically in the outer tissue of certain sponges and coleotrates and molluscs plus worms. Yes, these algae cells release oxygen during photosynthesis, which is beneficial to the host. It written algae gets suitable matrix for growth and the nutrition from the animal. Clear? So we having also a table, that is the parasitic protozoans, their names of the diseases, causes, organism and the vector. Clear? Yes, I hope you understood. So here some points to be remembered. You can read by your book, and this table also very important. Okay, thank you.